DocTalk is brought to you by Merck Animal Health. At Merck Animal Health, we're shaping the future of animal health with pioneering science, connected technology, and insights-driven solutions to bring our customers an unparalleled portfolio of choices to improve cattle care and operational efficiency. We support you and your legacy by helping you meet the challenges of today with the innovations of tomorrow.
Hey folks, welcome to DocTalk. I'm Dr. Dan Thompson. Thanks for joining us today. Dr. Aspen Workman, Dr. Vander Ley are going to join us here in Lincoln, Nebraska. We're going to talk about BVD, we're going to talk about gene editing, we're going to talk about BVD resistant cattle. Thanks for joining us. We'll be right back.
When you spot BRD in your cattle, that's your golden opportunity to target infection and its associated fever with a single dose of Resflor Gold, the industry standard dual therapy. To learn more, talk to your Merck Animal Health rep or your vet and see label at resflorgold.com. Animals intended for human consumption must not be slaughtered within 38 days of treatment. This product is not approved for use in female dairy cattle 20 months of age or older, including dry dairy cows.
Hey folks, welcome to DocTalk. I'm Dr. Dan Thompson and I am here with Dr. Aspen Workman and Dr. Vander Ley, and you can see behind us we are here in Nebraska at the University of Nebraska where this is Husker country, right? Dr. Vander Ley is an associate professor at the Great Plains Veterinary Education Center here in Nebraska. Dr. Workman is a research microbiologist at the USDA Meat Animal Research Center in Clay Center. And, you know, it's just been an unbelievable working together between the University of Nebraska and USDA in Clay Center between GP Beck and Mark, right? Those who everybody knows, Mark and GP Beck. And this is really fun for me because the work that you've done and what you're doing is going to change our industry. I know it, and very thankful. And so we're going to get into this and talk about some of the things they've done with genetics to produce a BVD resistant bovine, and it's incredible. But Brian, kind of walk us through why BVD is bad.
Yeah, BVD has been a problem for a long time. BVD is one of the most important viruses that we fight with in cattle because it's kind of got a bad name because it's bovine viral diarrhea virus, but it actually is a highly immunosuppressive virus. So when we think about respiratory disease in feedlot cattle or pneumonia in young calves, that's a big driver, that immunosuppression that BVD causes. So we've been working on it for a long time. The first reports have come out over a century ago, and there's been a long history of people in veterinary medicine trying to find solutions for BVD that whole time. So we see a lot of vaccine use and a lot of testing, a lot of expense, and then the estimates of economic loss associated with BVD infections in feedlot cattle are significant.
Yeah.
Even worse when cattle are as valuable as they are right now. So
yeah. And kind of the Trojan horse, right, is that persistently infected calf. And how does that interplay with this?
Yeah, so the persistently infected calf is a unique thing. We don't see that happen very often. In fact, I'm not sure it happens anywhere else in nature, does it? So these calves get infected when they're in utero, they're in the cow shortly after the cow gets pregnant, and that calf's immune system is trying to figure out what's self, what's me, and what's outside. And at that point, the immune system is getting rid of all the stuff, all the parts of the immune system that are going to react against that calf itself. If BVD shows up right then, BVD gets recognized as part of the calf, and the calf can never get rid of it for the rest of its life, and they turn into the typhoid Marys. They shed lots of virus out of essentially every secretion, everything that comes off of or out of that calf is full of virus.
Yep. And you said that our current tools today to kind of take care of it has been vaccination and testing, right?
Mm-hmm. Yeah, so the testing and vaccination are successful. They work. They have challenges associated with them. One of my favorite USDA NOMS studies shows that about 4% of U.S. cow-calf producers test for BVD infections, but 67% of them actually vaccinate for them, which tells me that most producers view it as a problem their neighbors have. So,
Right. Yeah, exactly. And, that means a third of them aren't getting vaccinated.
Right.
And then those are the ones that come to my feed yards. Anyway, let's take a break. When we come back, more with Dr. Workman and Dr. Vander Ley. We're here at Nebraska. We'll be right back after these messages.
DNA Dialogue is brought to you by Identity Beef, powered by Neogen.
Beef producers are subject to a lot of risk and volatility in their operations. When we think about the longevity of an operation and the profitability of a modern beef operation, it's largely dependent on the productive life and the economic drivers behind the cow. Oftentimes, a genetic condition or a genetic mutation corresponds to an undesirable phenotype or a calf with a negative economic value. That calf is not as profitable or has some sort of characteristic that is discounted amongst the rest of the animals in that calf crop. Genomic testing allows the cattle producer to determine whether that undesirable phenotype is caused by an environmental or managerial condition, or if it's controlled by certain genes or certain variants. When a producer tests their bull battery for genetic mutations, it allows them to manage problems before they even exist.
One of the most core principles in the beef business is reducing risk. We have eliminated and reduced the risk of animals not performing up to our expectations through the use of EPDs, through the use of genomic testing. I've gravitated toward the Angus GS, using it. You've got all the right people in the room when you've got Neogen and AGI working together. That's what puts my confidence in the Angus GS test. Cattle producers have always been sustainable. We have no choice but to be sustainable. But however, this is another tool to identify the most efficient animals, which take less inputs, less resources, and produce more beef with less inputs, less resources than ever before. And I think the evolution of genomic testing is vital to the cattle business.
Angus GS, powered by Neogen, is the highest industry standard in genomic testing for your whole herd. Contact the American Angus Association or your Neogen territory manager to test today.
Bovolus Nasalgen 3 offers young calves unrivaled protection against devastating respiratory diseases, including IBR, PI3, and BRSV. And it has a unique blue shadow, so there's no second guessing which animals have been vaccinated. To up your protection, choose Bovolus Nasalgen 3 PMH, the first and only intranasal that protects against viral and bacterial pneumonia. Talk to your veterinarian and visit nasalgen.com to learn more.
When the ones who have your heart need your help, count on us for everything they deserve, delivered fast. We'll work hard for you so you can work hard for your dreams.
Hey folks, welcome back to DocTalk. Dr. Dan Thompson here at the University of Nebraska at Lincoln with Dr. Brian Vander Ley, who's an associate professor at the Great Plains Veterinary Education Center, and Dr. Aspen Workman, who is a research microbiologist at the USDA Meat Animal Research Center. And kind of talk me through what the terminology and the processing, this gene editing, genome editing, what does that entail?
Right, so this new technology that's become recently available is called gene editing or genome editing, or you can sometimes hear it called CRISPR editing.
Sure.
And really, this technology is really exciting because it allows us to make very specific targeted changes in an animal's genome to try to get a new and desirable phenotype. So BVD, kind of like all viruses, needs to get inside of a cell in order to replicate itself and cause disease. And the way that the virus gets into the cell is that it recognizes this specific protein on the cell surface known as a receptor. And this receptor kind of serves as a doorway that lets the virus come inside of the cell, so it has access to all the resources of the cell in order to replicate itself. And so it's like the virus has a key. So it interacts with this protein, it can unlock the door and get inside of the cell. In our case, we were using this new gene editing technology to make a very small, precise change in CD46, in the gene that encodes that protein, so that the protein is still expressed on the cell surface, so that door is still there, that door is still functional, but it's like we've rekeyed the lock. So now when the virus tries to interact with that protein, it's like it doesn't have the key, and so the virus can no longer use that doorway to get inside of the cell and cause disease. So we're trying to use this technology to make a very, very small, precise change in a single gene to effectively block BVD infection in cattle.
It's unbelievable. Okay, so when you say we're doing something with the genotype to have a phenotype, you're saying you're doing something with the genetics inside with the cells to have a desired outcome or appearance or immune response, right?
That's absolutely correct. Yep.
And so I guess one of my questions is, because this sounds, I mean, it's, how long did it take to do something like that?
So in our case, we already knew which was the receptor that was required for BVD infection, and work had already been done they knew the exact residue, so the specific place on CD46 where the virus interacts. So that took years of research before we even came into the picture. And then we collaborated with a company that specializes in gene editing. So we collaborate with the company Excelligen. And then the actual process of making the edit, once you know the target and everything, doesn't take that long. We can make an edit in a cell in about a month, but then you have to take that through producing a live animal, and so that can take.
So one of the questions then, obviously, is this gene deleted in the wild already? Are there some animals that have that, or is this just something that none of them have it?
So what's special about this is that we can use gene editing. In conventional breeding, what you want has to already exist in the population, right? And with gene editing, it doesn't have to already exist. You can introduce something that doesn't exist in nature, and that's what we did here. This is a change in that one particular gene that doesn't normally exist.
So then, if this animal or animals that you do that to propagate, then they could pass that genetics on to the next generation, or potentially.
Yep, that's right. So they have this change in every cell in their body, and so it is heritable. It will pass on to the next generation.
It absolutely blows my mind. And folks, if you haven't ever looked at what they do at the USDA Meat Animal Research Center and Great Plains Veterinary Education Center, I mean, this is the hub of animal health research in the United States and the world. And it is an absolute gem for all of us to be proud of. And for y'all to do this for us, for our industry, for veterinarians, just remarkable work. So when we come back, we're going to get into talking about specifics of the animal that they produced that is resistant to bovine viral diarrhea. You're watching DocTalk, and we'll be right back.
When it comes to treating BRD, you want a product that you can count on to get the job done at an affordable price. Macrocin by Bimeda delivers on both. A straight shooting, no BS tulathromycin that does what it's supposed to do. End of story. You don't need to take our word for it though. Go to macrocin.com for customer testimonials and head-to-head trial results. For your cattle and your bottom line, choose Macrocin.
In the livestock industry, castration is a common practice to ensure proper herd management. All methods are painful, regardless of the age of the animal. At Solvit, we could not ignore the clear industry need for better castration solutions. So we developed LidoBand, a novel lidocaine-impregnated elastrator, addressing the pain associated with band castration. It provides local anesthesia throughout the castration process. LidoBand, a small device that can make a big difference.
When the ones who have your heart need your help, count on us for everything they deserve, delivered fast. We'll work hard for you so you can work hard for your dreams.
Hey folks, welcome back to DocTalk. I'm Dr. Dan Thompson with Dr. Brian Vander Ley and Dr. Aspen Workman. Dr. Vander Ley is Associate Professor at Great Plains Veterinary Education Center. Dr. Workman is a research microbiologist at the USDA Meat Animal Research Center, and we are in Lincoln, Nebraska. As you can see behind us, we are at the University of Nebraska. Just a beautiful facility, beautiful campus, unbelievable curriculum. So you get a chance, swing through Lincoln. A lot to be proud of here with what y'all do for animal health, production, animal performance, meat, everything that has to do with cattle. UNL has been central to a lot of it. So we want to get into talking about the production of this animal that is resistant to BVD, this heifer. So talk about, where did it start? You talked about the CRISPR and the gene deletion, but then after after you had that figured out, what were the steps that you did to show that this was going to work?
Yeah, so we really took a stepwise approach in trying to see if this edit would really protect first cells in the laboratory and then a live animal from BVD infection. So we started by just taking cells that are typically grown in the laboratory for studying BVD, and we first made the edit in those cell lines. And so then we can characterize what is the impact of this edit on susceptibility to the virus in the laboratory.
Yep.
Because then we can test a wide number of virus isolates from BVD. There are two main genotypes of BVD. So in the lab, we can test these cells against both genotypes of the virus, against all the different subtypes of BVD that circulate in the US, and begin to kind of characterize what's the impact of this edit. And then once we felt really comfortable in the cells in the laboratory that this edit is giving a phenotype that we expect and that we would like to transfer to an animal, then we took this technology to try to apply it to creating an animal that actually has this desirable edit in it.
And what were the next steps, Brian? You got that?
There's a couple of really important things when we do any kind of genome editing like this. We want to make sure that we don't create unintended consequences. So there was a tremendous amount of effort put in by some of the other members of the team to make sure that the edit happened exactly where it was supposed to happen and that there was only one. We didn't want to put a bunch of copies of the edit into the genome of this animal, because we would get other kinds of effects if we did that. So that edit was confirmed through whole genome sequencing. So they looked at Ginger, the animal out here, from one end of her genome to the other, made sure that the edit was in the right place. It was on both sides of that DNA strand, so both copies were good, and then that there was only one. So after that, then the next part of the process to see if we can get a live, healthy calf. And that part it's pretty interesting. There's a lot of reproductive technology that goes into gene editing. There's the whole gamut of things that we would do in normal bovine production today anyway. There's technology like IVF and cloning are pretty common, even in commercial animals. So a lot of that stuff has to happen. And we ended up with an animal that is healthy by all the measures that we can get. And she's doing really well, even today, as almost a 2-year-old.
To take the stepwise approach in vitro and then produce a calf, that's a lot of work, folks. And so thank you for explaining that. When we come back, we'll talk about some of the tests that you put Ginger through, right, to make sure that it worked. And we'll talk about the future of this technology. Incredible stuff. Dr. Vander Ley, Dr. Workman, we're at University of Nebraska-Lincoln. We'll be right back.
The cost of an open cow these days is very expensive. It's hard at times to dedicate a half a day or a whole day to leave your practice to go palpate cows. And so Alertis Test has helped speed the process up of getting preg rates back to the owners. And so free up time for diagnostics or working in your clinic. It also will help generate revenue, not being on site to do the testing.
When a new calf hits the ground, the clock starts ticking. A belly full of colostrum gives them their best chance, but if they don't get any, time starts running out. That's when you grab a bag of Oxford Ag Colostrum in their patented feeding system. It's simple. You fill it with warm water, shake it to mix, feed it with a tube or a nipple, and you are done. No bucket, no bottle, no mess, and right on time. Ask for it by name wherever animal health supplies are sold.
When the ones who have your heart need your help, count on us for everything they deserve, delivered fast. We'll work hard for you so you can work hard for your dreams.
Hey folks, welcome back to DocTalk. Dr. Dan Thompson here with Dr. Brian Vander Ley, Dr. Aspen Workman. Dr. Workman is a USDA meat animal research microbiologist, and Dr. Vander Ley is associate professor here at GPVEC, Great Plains Veterinary Education Center, associated with the University of Nebraska at Lincoln. If you've been to the Academy of Veterinary Consultants or been at NCBA, you've seen Dr. Vander Ley speak all across this country on bovine health, and the research that they're doing together, just really, really incredible. And we're very thankful you took the time to spend with us today. Let's talk about what we did with Ginger then, after she was born, and how you tested to make sure that she was resistant to BVD.
Yeah, so I can address that a little bit. So just to clarify, I don't think we've talked about this yet, but Ginger is the gene-edited heifer that has the edit that changed the lock on the door for BVD. So BVD should have a hard time getting in, but we wanted to make sure that, Dr. Workman did a lot of work in the lab to show that BVD really does have a hard time getting into these cells. It worked really well in the lab, but the proof has to be in the live animal if we want it to work in the field. So we needed to do a live animal challenge. There's a lot of different kinds of challenges that have been done out there. You can do really simple ones where you just expose them at one point, just put some virus in their nose or something like that. But we wanted this one to be real life. So we got a persistently infected calf. And we actually, they were in a room, like a pen that was relatively small, and we put the calf in with Ginger and the control animal. And we actually have some really good footage of the challenge taking place because there's a lot of nose-to-nose contact. So we were confident that they were exposed.
So when an animal is first exposed to BVD, the virus replicates in the upper respiratory tract and then makes its way into the blood. And the blood carries BVD throughout the body. And in the blood, BVD infects and kills the white blood cells. And so we looked for the presence of BVD in the blood of Ginger and then our just normal wild type control calf to see, is the virus making it into circulation? And what we found is that in Marianne, which is the name of our just normal wild type control calf.
I get it now. Marianne and Ginger. I'm slow, but
we could detect BVD in her white blood cells for about 2 weeks after the exposure. We could detect virus in her blood. Whereas for Ginger, we could never detect infectious virus in her blood, and her white blood cells remained uninfected during the entire course of our challenge study. So that has huge implications for controlling BVD and the spread of BVD.
it's unbelievable. It's so refreshing and so cool. So what's next?
I think the big next step, the big next question is that most important infection happens in that calf before it's born.
Yep.
And we have some plans to do some research to look at challenging heifers or cattle that are pregnant, carrying calves that are edited to find out if that edited fetus can protect itself from the virus infection. We also, we have one animal. That's one of the consequences of doing the type of work we had to do. So it's really important. We want to make sure that before people spend a lot of time and money adopting technology like this, that we have thoroughly assessed it. So we actually have a group of calves that are gestating right now. I think they're Angus calves. They're going to look a little different than the one we've got today. And that we're going to redo the challenge. We're going to make sure that it works a couple of times, and we're going to do it with a few more animals. But I think the key thing is to make sure that that edit will protect the calf in utero, to make sure that we break the chain of making new PIs, which is really where the leverage to stop BVD is.
Absolutely.
So,
well, thank you both for taking time out of your schedule. Thank you for being on the show. But most importantly, thanks for working together and bringing something to the industry that is just off the chart exciting.
Thank
Thank
you.
you.
Yeah.
Thanks for having us.
So thank you for watching DocTalk. This has been maybe one of the most interesting shows I've ever got to do, and just a lot of fun here at the University of Nebraska. Thanks to the USDA Meat Animal Research Center. Thank you to Great Plains Veterinary Education Center. Thanks for watching. If you want to find us on the web, you can find us at www.doctalktv.com. Always work with your local veterinarian. And I'm Dr. Dan Thompson with Dr. Brian Vander Ley and Dr. Aspen Workman, and we'll see you down the road.
At Merck Animal Health, we're shaping the future of animal health with pioneering science, connected technology, and insights-driven solutions to bring our customers an unparalleled portfolio of choices to improve cattle care and operational efficiency. We support you and your legacy by helping you meet the challenges of today with the innovations of tomorrow.